page.
No, it's live on Facebook. It says in the top left corner. Now it's streaming live on Facebook.
Yeah, I think I got it though.
Okay, so you got it?
Mm-hmm.
All right, good.
And it's working on my phone. So there it is.
Hey.
There we are. There I am multiple
Don't
times.
we love technology? All right.
Sweet.
Sorry about that.
My apologies for the unexpected technical difficulties. We shall begin. Whoop, and I ended up on the second slide. Good evening, everyone, and welcome to our June 15th meeting of the Western Pennsylvania Mushroom Club. My name is Stephen Buckland. I'm the current president, and I've got some updates on our club's activities over the past month. Everyone should have received their May/June newsletter. There's a lot of articles in there, including some updates on Dye Day and our annual Linkoff Memorial Foray. There's also a really fun article by Richard Jacob on eyelash cut mushrooms. I really enjoyed reading that. So if you're into microscopy and tiny mushrooms and things like that, check it out. If there's anything you'd like to see in the newsletter, submissions are always welcome. And you can submit things to our editor, Cecily Franklin. The next issue of the newsletter will be in your physical or electronic mailbox on July 1st. So look forward to that. Since our last meeting, we've had 4 in-person walks. First up was May 23rd at Barking Slopes. This was hosted by Allegheny Land Trust. Julie and myself were the identifiers on this walk, and our coolest find, in my opinion, was a rust fungus growing on Jack in the Pulpit. It's called Euromyces aretriphyli, I think. That's the picture right there. Super cool little rust fungus. Next, there was a walk with Richard Jacob as part of the FIPS BioBlitz. It was a hot afternoon and it yielded a lot of your typical late spring, early summer mushrooms. And Richard wrote up a really nice thing on our website about this cool fungus, which is Mycoacia uda, pale yellow splash tooth. And when you put a drop of ammonia or potassium hydroxide on it, it gives you this purple reaction that you see over here. I was really fascinated by that find. And Richard says it's only been reported once before in our area, and it hasn't been identified at a club walk prior to this. So pretty cool find. Then we had a walk 3 days later at Gertie's Woods. This is a new Allegheny Land Trust property in Etna, which is right across the Allegheny River from us. And Kara and Julie were identifiers for that walk. And yeah, if either of you want to jump in and share anything, feel free.
Sure. Yeah, it was great. It was the first time that we did a walk there, and so we didn't know any of the trails. Everything was pretty much new for everyone, except for maybe Julie. I think you were there for a different kind of walk, right?
Nope.
No, you weren't even
there.
But I was so excited to see the blushing rosette. It's this beautiful mushroom you see on the right there. That was the first time for me seeing it. And it just, it has, when it's younger and it's forming, it gets this beautiful red gutations, that liquid that exudes from it. And it was such a pleasure to see. We saw a bunch of good stuff. Mm-hmm. For such a, I mean, it was quite disturbed, but even for as disturbed as it was, we found some really good stuff, and it was a good time. A lot of people showed up.
Yeah, I was pleasantly surprised. Lots of good mushrooms there. So Stephen and our walk at Barking Slopes was kind of like a crust and rust today because it hadn't rained in like weeks, I think. So it was nice to actually find some some cap and stem and some legitimate big mushrooms. So
Squishy ones.
yep.
Awesome. Next up was a walk at Beaver Meadows up in the Allegheny National Forest. And yeah, Kara, anything you wanted to share about our finds from that day?
That was a blast. That was my birthday walk. I'm 40, everyone. So yeah, so this was my birthday walk. It's one of my favorite places in the world, Beaver Meadows. Always do well for mushrooms there. As you can see in the first picture here, you see Barbara taking a photo of this huge conch right there, Fomatopsis ochracea. And we had John Plischke actually lighting it on fire to see if it bubbled on the edge, to see if it was Munsii or ochracea. If it didn't bubble, it was ochracea. If it did bubble, it would have been Munsii or Muncie. I can't say Latin, forgive me, but it's the one with the M. Anyway, so then we got two Vivaceae species. Ugh, the one that grows on the trees, the truncorum, the Vibracea truncorum, and then the one that grows on leaves, which is Vibracea foliata, something with an F. It's the other one, guys. I'm such a great identifier. And then as you can see here, we have one of my favorites. It's not like the most uncommon thing in the world, but I just love finding it every time. The Entolona incanum, the mouse pea pinkill. And yes, if you smell it, it does smell like mouse pea. And it is beautiful. If you pick it up, it stains blue. It's just gorgeous. And as you can see, everyone on the ground here, that's because we were all finding cool stuff. A lot of them were all getting down for those entolomas that we found. And then there was something in the back there that Garrett and John found. They were fighting over the ID. So I'm not going to take sides on that one. But not fighting, but you know what I mean. And then we have some Matrula. I did scope it, and it was all Matrula elegans. So yeah, good old Matrula. A lot of good stuff we found there. It was a lot of fun. Some boletes. I had a blast. So much fun with everyone there.
Yay, yeah, I'm so glad we had so many walks in the past month, and we've got a lot more walks coming up. So upcoming walks with ALT, we've got the Beginner's Walk at Bethel Green on July 16th, Sycamore Island on the 31st, and then Churchill Valley Greenway, which is another new ALT property, and that'll be on August 20th. And there'll be more as the year unfolds. These are just the ones in the next couple of months. And registration for those is on Allegheny Land Trust website, and registration is limited. And just for limited registration walks, please only register if you plan on attending, and please contact Julie if you are unable to attend for these walks. We've had some issues with some low attendance on sold out walks. So be courteous, be kind with your registrations. Next up, we've got some other walks, some happening much sooner than July. We've got June 19th, the Dorothy Fornoff walk at Deer Lakes Park. And you'll meet Richard Jacob at 10 a.m. at the parking lot between the first two small lakes. And there's no pre-registration for this, just show up in the parking lot at 10 a.m. June 20th at McConnell's Mill State Park, Jerry Sapp Jr. will be meeting folks in the Kildew parking lot at 9 a.m. And you'll get to go on a beautiful walk under the hemlock forest there along Slippery Rock Creek. It's a true gem of Western Pennsylvania if you've never made the trip up to McConnell's Mill. And then July 17th, one of our new identifiers, Barbara Badakova, will be leading a walk at Heartwood Acres and meeting at 10 a.m. tentatively at the mansion parking lot, but there may be construction limiting our access to parking on that side of the park. So stay tuned if you're interested in that walk. We'll be updating the meeting location probably as we figure out some stuff. Yeah, and for those of you who are perhaps vaccinated and travel inclined, this year's NAMA foray will be at the Snow Mountain Ranch in Granby, Colorado. So there's some details there on that event. Our NAMA trustee, Garrett Taylor, will be attending that foray, and maybe some other club members have an interest in going. In terms of our upcoming meetings, we have tonight's meeting and next month as our last two virtual meetings of the year. And then we'll be resuming in-person meetings on August 17th with our annual cultivation meeting. We will begin log drilling for shiitake cultivation at 6 p.m. So if you have a recently downed oak log or another hardwood log and you want to get that inoculated with some shiitake spawn, definitely recommend showing up early. The drills will be starting at 6 p.m. So yeah, then the rest of the year, hopefully we will be in person at Beechwood Farms in Fox Chapel, just like we used to be. Crazy. All right, other upcoming events. We've got Dye Day on Saturday, August 21st. The walk in the morning is free. The afternoon session is $10. The walk, I don't think has been decided on where we'll be meeting for that walk, but we will be taking mushrooms from the walk back to Harmer Shelter to identify them, and then we'll probably have lunch there and pack your own lunch, and then the afternoon session will be learning how to dye with mushrooms. So that'll be really awesome. We'll also have some books for sale. We're ordering some special books about dyeing with mushrooms, and we'll have those available for purchase if you're really into it and you want to keep practicing that on your own. And then last thing, to register for Dye Day, if you go to our events page, so wpamushroomclub.org/events, and scroll down and click on the listing for Dye Day, that'll take you to a form where you can input your name and pay the $10 registration fee. And last but definitely not least, I've got some info here on our Gary Linkoff Memorial Foray. Registration is now live on our website. Fee for the main event is $60 for non-members, $40 for members, $25 for students, and free for children 10 and under. We'll also have Science Sunday this year from 9 a.m. to 4 p.m. It'll be 3 to 4 workshops covering topics such as microscopy, photography, and mushroom identification. Registration is required and it will be $20. And of course, there's the pre-foray walk at Cook Forest State Park, another Western Pennsylvania gem, and that one is free. So just show up and join us for a walk on Friday. Our speakers this year are Leon Chernoff from Mushroom the Journal and Michael Kuo of mushroomexpert.com. Leon's talk is titled Life as a Mushroom, A Mushroom's Eye View of Life. And Michael's talk is titled This Lecture Stinks, Fun with Phalloid Fungi. So I think those will both be really engaging and interesting talks. I'm really looking forward to it. They both seem like people who have a good sense of humor, so I'm sure we'll have some laughs during those talks. Here's a membership report. We are up to a record 1,099 members as of today. So we've gained 184 new members so far this year. And yeah, I just wanted to say thank you so much to all of our members, new and old, for supporting the club. And it's an honor to serve so many mushroom enthusiasts from Western Pennsylvania and beyond. So thank you all. And if you're watching the recording of this and you're interested in joining our club, dues are $15 for an individual, $20 for a family, $10 for a student. You get all of these benefits listed here, plus you get our bi-monthly newsletter, updates on mushroom walks and events. You get a discount on your registration for our annual Linkoff Memorial Foray. And if you can attend the in-person August meeting, you get a mushroom grow kit. On top of our shiitake log cultivation, we also do oyster mushroom kits. So a lot of benefits to becoming a member. Next, I wanted to share some Mushroom ID Awards. Our Button ID program recognizes club members who can identify different species of fungi. Buttons are awarded at the 10, 25, 50, 75, 100, and 150 level, and maybe higher. I don't know. Julie will have to let you know if there are other buttons. But members can submit their list for verification to the email listed above, and you can generate your list fairly easily. If you post your stuff to iNaturalist or Mushroom Observer, you can get that to make a list for you, or you can just keep track of it yourself. And yeah, check out the Button ID page of our website for some more details on that. So tonight we have 4 members we're recognizing for their identification efforts. First up, we've got Eli Nitzberg and Heather Cole, who both submitted their 10 species lists. And yeah, I think I'm going to turn things over to Julie, who is our Button ID chair.
Yes, I love looking at everybody's lists, even on our beginners 10 lists. Sometimes you have mushrooms I don't have, so it's been really cool just to look through everybody else's observations and what you're seeing. And I'm really excited because once you get 10, it's all up, it's all good from there. You start earning more, and we do have over 150. So I believe we have a 200 and a 300 button. There might even be a 500 if I root in the box deep enough. There is probably a 500 button lurking in there. Or I could make one, but it will take me a while to get through a 500 list, and I will probably make many other people look at it too. But yes, please send me your list of mushrooms so I can get you your button. So congrats to our 2 newbies here, Eli and Heather, on getting their first of hopefully many buttons. If you do send your list to me, that email forwards directly from to my work email. So you will get an email response from Allegheny Land Trust email address. So just know that the message coming back to you will be an Allegheny Land Trust email. Easier to keep track of my emails that way when they all go to one. BA, Heather, and Eli.
Woo. And then we have 2 more folks we wanted to recognize who are at the 150 and identifier level in the club. So yeah, Julie, go ahead and share the other awards.
Yes, super jazzed to have Barbara and Beth join. If you don't follow Beth on all of her journeys, her mushroom list was incredibly impressive. Same with Barbara. Barbara has some mushrooms from all over the world. Oh my goodness, devil's tongue was on there, and some mushrooms just from all over Europe. So that was really cool. Barbara's Instagram, Fungi woman. Is that right, Barbara? Fungi woman, amazing, amazing photos. So if you want to get some awesome photos and info, check out her Instagram. But congratulations, super excited to go to you guys' walks that you're gonna lead. And I think we've got at least one other person coming down the pipeline for 150. So that'll hopefully be at our next meeting. We can announce that. So congratulations.
Yeah. So awesome to see more folks becoming identifiers for the club. So I just want to remind folks that if you're interested in learning to identify more fungi, whether it's to earn your next mushroom ID button or purely for joy, stick around after the speaker for our mushroom ID session. So this is a space we created so that folks could bring pictures of any fungal finds, whether it's just to share and have conversation or to get some help identifying them from our expert identifiers. Yeah, it's always great to have folks stick around and share what they've been seeing out in the woods. And then last announcement, I think, is a huge thank you to Carissa Mendez, who has volunteered to step into the role of hospitality chair. And you'll be able to see her debut at our August cultivation meeting. So thanks so much, Carissa. We really appreciate you volunteering for the club. All right, so next month's meeting is going to feature Thomas Roll. He is a WPMC scholarship grant recipient, and he's going to be presenting his research on Flammulina veludipes. Thomas is pursuing a master's degree in biology at the University of Wisconsin-La Crosse, where he is advised by Todd Osmundson and Tom Volk. So, yeah, that'll be another fun speaker. Any other questions or announcements before I introduce tonight's speaker? I think I can change this so that yeah, now people can unmute themselves. So if you have anything you want to share, you've got a couple seconds. I think I caught up on the chat here. Someone asked about live streaming our in-person meetings. We'll definitely be trying to do that. I just shared that it might be kind of hard for us to stream the cultivation meeting just with all of the drilling and stuff going on. But definitely our future in-person meetings will continue to live stream onto Facebook and put recordings on our YouTube channel. And I think I heard someone unmute. I didn't know if there was a question, but if not, I shall.
I have one.
Oh, yeah.
Does the Gary Linkoff foray have lodging possibilities?
It's not built into the foray, but we can definitely help you figure out lodging.
Got it. Okay, so just look at camping stuff on my own.
Yeah.
Okay.
There's probably a member that'll let you crash with them too.
Yeah.
Okay.
Do a little couch surfing.
Cool. Thank you.
You're welcome. Well, I guess I will introduce you now, Kristen.
Okay.
So tonight we welcome Dr. Kristen Wickert, who is a forest plant pathologist who specializes in the fungi of our eastern forests. She's going to highlight some of the obvious and not so obvious fungal players that are influencing our forest composition. Dr. Wickert has a bachelor's degree in forest biology from Penn State, as well as master's and doctoral degrees in forest plant pathology from Western West Virginia University. On the topic of plant pathology, Dr. Wickert says it's like a giant puzzle with many different pieces that tell a story about our ecosystem's health. Fungi are commonly an important piece in that puzzle. And yeah, so with that, I welcome Kristen and turn things over to you.
Thank you. Your club is very impressive. It's very organized and you have a lot of members. It's really inspiring. I think it's good to go. Okay. So I love questions. However, I get sidetracked. So if you could hold your questions to the end, I will gladly talk forever about this stuff at the end of the talk. But like I was introduced, we're going to talk about fungi as plant pathogens. And a little bit more about me. I'm very blessed to be someone who works in a lab and also someone who gets to work outside. And my official title is very long and silly, but we'll call it a forest plant pathologist. And I have the bachelor's in forest biology, which was very broad at Penn State. And it was different than just getting a forest management degree, which I still did the chainsaw stuff, but I also did like conservation genetics and all sorts of things that included like mycology. Then I went on to get a master's in plant pathology that focused on entomopathogenic fungi. And it sounds different, but just wait, you'll see how important insects and entomology are in plant pathology. Then I got my PhD in plant pathology and I studied solely plant pathogenic fungi in the works in the ecosystem around Pennsylvania. And I've also spent time working for the Forest Service as an entomologist and also as a forester, where I did urban forestry. I did, you kind of name it. I was everywhere. And it was a lot of fun, but I definitely enjoy research and learning. So I have lots of years and lots of debt from school. But now today I am a forest entomologist and forest plant pathologist. And I've written or contributed to a lot of different papers talking about the interactions of fungi and insects, fungi and plants, and solely just plants and solely just fungi. So if you have any questions about anything today, please ask, and there's a good chance that you can steal some of the knowledge from my 10 years of school. But what is the field of plant pathology? That is the study of organisms and abiotic stressors that cause diseases to plants. And there are a lot of things that can hurt our plants. So there's a lot of people who have to specialize in studying those distinct things that can hurt our plants, such as nematologists, which are little tiny round worms that you have to use a microscope to see, bacteriologists who study bacteria, and of course, mycologists who study fungi. And even within plant pathology, there are a lot of different types of systems we work in. So I specialize in mycology, plant pathology, in forest plant pathology, meaning that I am, as my mom says, a tree doctor, but really I mostly kill trees. But there are plant pathologists who study soy. There are a lot of them. There are a lot of people who study corn. It's a big agricultural thing that you pay for with your taxes. And it affects your life in way more ways than you would really want to acknowledge, I think. But I work in the forest and I study fungi that affect hardwood and softwood trees and try to keep them from dying. And like I said, a lot of being a plant pathologist is making observations and also killing plants. And there's a famous mycologist, well, he's actually a microbiologist, Robert Koch, and he is deemed with making the process that all plant pathologists use to prove a point called Koch's postulates. And in order to say that an organism is causing a disease, you have to first use your special eyes and make an association where you observe a plant and see that it is not healthy. So you can notice things like this chlorotic tissue, the yellow. You can also see things like wilting. You can see necrotic tissue, which is this brown area here. And sometimes you'll even see some sort of a sign of an organism itself, like these black dots. But in order for you to say that that organism or association is happening to that plant, you have to pull out the organism that's causing that disease. And so we do that in mycological plant pathology by using agar plates. And what an agar plate is in my hand there in that picture is literally just sugar, potato starch, and it's agar from seaweed. So it makes the perfect environment for if you were to take fungi out of the plant it's living in, it'll say, whoa, this is way easier for me to eat, and it'll grow out, and it causes these beautiful fungal colonies, which would otherwise be hidden from view. So you isolate these causal agents, and then to confirm that that fungus, bacteria, or whatever is causing the disease, you put it back into the same type of plant, so same species and sometimes even the same variety, and you see if you get the same exact symptoms, and if you can pull it out again out of that new plant to get that same causal agent. So that's a lot of what being a plant pathologist is trying to find out who is causing what and confirming it so other scientists can know how to control for diseases. And here's a real life example in Pennsylvania. So this is the plant pathologist at Penn State. If you ever have a question and Penn State Extension doesn't know, they'll call this guy, Dr. Don Davis. He was a very important scientist in my life at Penn State, got me into mycology. He also is a lichen expert. So if you like the fungi and lichens, you can contact him. But there is a parasitic plant in Pennsylvania. It's actually a shrub, and it's what he's holding there. It's called buffalo nut, Pyrularia pubera. And it was noticed that that plant was showing signs of disease. It was showing these distinct cankers. It had sprouts coming up from dead and dying stems, and it also had these weird pustules on the stem. So if you remember, we have to say there's an association, then you have to take out what you think is causing the disease on those agar plates and put it back into the plant. So that first step is association. See that yellow dotted line lasering in on the association of what's wrong with that plant. And then you'll perform isolation. And so you take chunks of the plant, literally with a scalpel, scoop it off. There's a lot of sterile practices to this. And you'll plate it into one of those agar plates. That's just sugar, starch, and a medium for it to grow in. And you can see several different types of fungi will grow out. So this pink colony here, that's a fungus. Same with this black and white colony here, which is also a fungus. And we can use microscopy skills to identify those individual species of fungi. And we can take that then when we have a pure colony and put it back into healthy plants and see if we get the same problem, which in Pennsylvania we did. So we took out the fungus that came out of this sick buffalo nut and put it into healthy branches in the lab, and we were able to get the same symptoms and the same level of disease. So that is what we do on a daily basis in the lab. And I know you're probably saying, where are the big, beautiful mushrooms? I want to see all the things that look like this. Well, almost all of the big mushrooms that you see in the forest are either in a mycorrhizal or saprophytic relationship with trees and plants. They're not necessarily causing disease. There are a few exceptions of basidiomycotin fungi that are plant pathogens, like armillaria. So armillaria tabescens and malea, which we call the honey mushrooms, are plant pathogenic fungi. And when we grow them in the lab, they keep that rhizomorph life form in agar plates, which is really cool and always super fun to grow out. But if you're ever on a walk and you see a downed log and you see these like weird rhizomorph things, which I have also in my hand, I literally got these today, they're everywhere. This is honey mushrooms. So this is the melanized hyphal structure of honey mushrooms that lasts over winter and waits for a new host to be stressed enough that it becomes a plant pathogenic fungus. Some of our polypores are also considered to be secondary pathogens, where they start to infect trees that are weakened and will then deteriorate the inside. And that's why in some of your guidebooks, you'll see heart rotter or sap rotter, things like that. They kind of specialized on sickened and stressed out trees. But many of the plant pathogens are in the phylum Ascomycota. which is different than the big, beautiful mushrooms in Basidiomycota, except for morels. And they can cause vast amounts of disease in our agricultural systems and individual forest trees. And like I said, most of the plant pathogens are ascomycotin, but there are examples of plant pathogens that can be in different phyla of fungi, and even the obscure ones. But there's a lot of diversity, and most of them have to be distinguished by microscopic features of how many asci or parathecia are in a certain area, things like that. So the ascomycotin fungi, you think that they're not common, but they actually are. They're very common. You might have some that's less than 50 feet from you, several different things, because cheese includes ascomycotin fungi. So our blue cheese and our brie have penicillium that encase them and also grow throughout them. You might have beer or bread or alcohol in your house, which has Saccharomyces cerevisiae, which is an ascomycotin yeast. Medicine in your medicine cabinet, if you're on antibiotics, you can thank ascomycotin fungi that have the ability to produce toxins that kill bacteria. And of course, the one that no one wants to admit, but the diseases on our body like ringworm or maybe like tinea on your back or your athlete's foot on your feet, that is also an ascomycotin fungus. They are the reason that our produce goes bad. Those little brown dots on bananas are actually an ascomycotin fungus, Calototricum muscae. And we just have general environmental molds like aspergillus that's growing in, you know, the ducts of your house just on random things. But of course, there's also plant pathogens, which could be on the plant right behind you, which is, so all of these things are in my house right now. Pretty interesting. But a lot of these plant pathogens are really cryptic because all you see is the symptom that's happening on your plant, like that yellowing or the browning or the wilting or even the death of the plant. And they can even be hard to identify when they come out of the plant because they have a very mysterious life cycle. So this picture down here on the bottom with the really black and fuzzy, oh, it went crazy, it's on a timer. But this plate here with the black and fuzzy fungus has a very distinct life stage, but it is the same species as these red and white plates over here. This one is just older. It's literally a month older, and it starts to make resting structures a lot like the hyphae of the honey mushroom. So it has melanized tissues that make it look very different. And you can use clues such as their microscopic morphology to tell that they are different species, and often different genera is a lot easier. So this is an example of Beauveria bassiana, the fungus that eats insects, and this is Fusarium with a very distinct shape. Okay. But plant pathology has been around ever since, well, before us in early civilization. It's really impacted us. So this is our wheat rust, which is caused by the fungal pathogen Puccinia triticina. And there's actually a Greek god dedicated to this because people would pray to it because it would cause giant famines in early civilizations. And the only way to stop this fungus was by murdering female virgins and animals. So it's very entrenched in our culture as humans. Another example of plant pathogens that will affect our existence is the ergotism of Saint Anthony's fire. And it's Anthony of Egypt is another one of his names, but it's famous for spiritual strength in the face of torment from the devil. And this is a painting of what people looked like in the Middle Ages, where they had a lot of things happening to their body, such as insanity, but also convulsions, gangrenous limbs, and hallucinations. And all of this terrible stuff that was very common was happening because of a fungus that was infecting rye. So it's called ergot of rye, and the fungus, which is a plant pathogen, is called Claviceps purpurea. And it replaces the seeds or the grains of rye with a sclerotia, which again is a very dark melanized tissue. And out of that grow these little stromata that produce spores. But these little sclerotia structures get mixed in with the grain, and they contain lysergic acid. And I don't know if anyone likes hippie stuff or the 60s, but lysergic acid is a derivative of LSD. So all these people were just tripping, and they were having convulsions because they weren't getting blood to their extremities or enough blood to their brain. And ergotism, it still occurs today, but we have made literal machines that can sort out grain that prevents mass hysteria. And there are sometimes outbreaks of claviceps purpurea, and it affects a lot of poor people in poor nations that can't afford really big sifters that can distinguish the weight of the grain or the sclerotia. So it's still happening today and still affecting lives. Of course, the Irish potato famine is a really famous plant pathogen, and it's technically not a fungus, but it is an oomycotin organism. But because it's confusing, people throw it in with fungi. But it is an organism that has hyphae, which is why people think it is a fungus, or people used to think it's a fungus. But it does have swimming spores. So it has motile spores that have cilia and other appendages. And Ireland was the perfect storm for a water mold infestation, a lot like the perfect criteria of growth for fungi, which is warm, moist, and like sometimes followed by like cold, moist. But the moisture caused a widespread sweep of this plant pathogen that forced people out of Ireland to America or other places to escape hunger. And you can see some of these dark places in the map of Ireland show 30% of the people there left or died because of a plant pathogen. So a lot of you might directly be affected by a plant pathogen. I know where I'm from in Eastern Pennsylvania, there's a lot of Irish people. So I'm assuming in Western Pennsylvania, there's still a lot of Irish influence as well. And that organism, its genus is Phytophthora. So Phytophthora infestans cause the potato blight, but it also still exists today, just like the ergot of rye, and it affects other members of the Solanaceae family. So potatoes are closely related to tomatoes, and they sound similar, so that's helpful, but also eggplants and tomatillos. And just because you don't see it on the news doesn't mean it doesn't exist anymore, because there are scientists who study this and try to adapt. And one of those famous scientists, which is really close to you and influences the tomatoes that you grow in your garden today, is a professor at WVU. So an hour and a half in a suburb of Pittsburgh called Morgantown, there is a 97-year-old man who still goes to work and studies the oomycotin organism that caused 30% of Ireland to come to America, because fungi, especially plant pathogens, have a very good ability to almost mutate themselves, right? They're evolving all the time because we make new varieties of tomatoes and potatoes. So they make themselves new varieties that can attack these susceptible types of tomatoes. So this professor, Dr. Gallagly, has worked endlessly to make tomatoes that are resistant to Phytophthora. And that brings me into talking about the very narrow branch of plant pathology that I study as a forest mycologist that is also a plant pathologist. And we're going to talk about the American chestnut, which is a very common story that people know because it, again, affects a lot of us. And it wasn't so long ago that American chestnut was a really big part of our American life. So there was an American extinction that was caused by a plant pathogen. And this is the story of the American chestnut, where American chestnut used to be trees that were as big as this in our forests throughout all of Appalachia, especially Pennsylvania. And of course, we logged a lot of it, but a lot of these trees died because of a fungal pathogen. But the wood was really important in our area because people would literally use it to build cradles. They would also use it to build their caskets, and they would use it to build their barns and literally everything. These trees also produce really tasty nuts that have a really high carbohydrate level and fat level. So they are super healthy for people to survive on over the winter, as well as animals, which people eat. It's a never-ending story. But these trees also produce nuts every single year, which is different than oak trees because oaks have like weird massed year cycles. So when we lost the American chestnut, we lost a lot of our biodiversity because a lot of things suffered after it. And we also lost a lot of our heritage. But the organism that killed American chestnuts goes by the scientific name Cryphonectria parasitica, AKA chestnut blight, which is the disease. And again, there's that gorgeous picture of amazing hardwood trees, and I wish we could see it. I'm so jealous of these people back in the day. But American chestnut used to grow throughout all of Appalachia. And you can see Pennsylvania, West Virginia, we are completely covered by American chestnut. It used to be 1 out of every 4 trees was an American chestnut. And in about 1950s, there was a plant pathogen, this gorgeous orange organism that was introduced and causes a canker. So it kills the living layer of the tree underneath the bark, and it annihilated the population of American chestnut throughout almost all of Appalachia. And you will still encounter some chestnuts that look like this. And that's because this tree has an ability to grow out of the root stock. So these huge trees had huge root systems that are still struggling and trying to survive by putting up these trees, but they get infected as well and they continuously die. And so American chestnut is labeled as functionally extinct because it can't get old enough to produce offspring, all because of a plant pathogenic fungi. And this has greatly changed the forest composition of the East Coast. As I mentioned, the nuts were really good for deer, all sorts of animals, for bears, squirrels, and then everything else that ate those guys. But also to, tickle your guys' fancy, I threw in this paper that I just found today, which is kind of old by now, but it is from it's actually from La Crosse, where Tom Voke is. And I'm going to go back for a second to talk about this picture here. Is it right? Wait. No, never mind. We're going this way. Sorry, guys. It's unorganized, but it's a really important part of the story. Okay, here we go. The range. Okay, so look at this range and see how Appalachia has all these American chestnuts. But over here, where La Crosse is, in Wisconsin, there are no American chestnuts. But because people loved American chestnut so much, they took it with them when they moved out west. So there's these disjunct populations of American chestnuts, like this population here in western Wisconsin. And so there's these really big American chestnuts there that still remain. And there was a student who studied the fungi that were associated with them. And some of them seem like they're common species, like we might have seen, this Lacaria species here. Lacaria amethysta is pretty common, but just imagine, again, 1 out of every 4 tree species through Appalachia was an American chestnut. So if this organism was associated with it, just think about how many mushrooms there were versus what we have today. And what are plant pathologists doing today to combat this historical disease? We don't just go out there and say, yep, there's a disease and walk away. There's a lot of things we can do to try to stop it. So scientists have tried to hybridize American chestnut to make a resistance hybrid, because Chinese chestnut, where the disease is from, are resistant, natural coevolution. So they'll try to breed American and Chinese chestnut to hopefully make something that's resistant. It doesn't always work, and it takes a very long time. There's also a virus that infects the fungus and makes it unable to fully infect the tree and kill it. And then there's these transgenic chestnuts, which are making everyone really upset in the news because it is a GMO. And the GMO, which I fully support because I was involved with the science and I know what's going on and it's not scary, it is probably going to be what brings back the American chestnuts into our Appalachian forests. But my research, or my involvement in research that happened with the chestnut blight, was putting a virus into the fungus that caused it to not be strong enough. A lot like when we get a virus, we get weak, we can't actually perform our duties. So this fungus can get a virus, and it wasn't able to actually break down the bark of the chestnut tree as fast, and the chestnut tree was able to fight back and survive. And this picture on the right actually is that place in Wisconsin that has the American chestnuts that were brought over, but they also, this stand has the virus. And these big trees, they still have cankers because of the fungus, but the virus has slowed it down enough where these trees aren't totally dead. But this virus research started in the 70s, and it's now just starting to kind of work. So a lot of these research things take a lot of time to understand and also to implement. Another example of a fungus that is wreaking havoc on your ecosystems in Western Pennsylvania, there's a disease called beech bark disease. And this is going to bring in why I'm an entomologist as well. It's the perfect example of why entomology matters in studying plant pathogens, because this disease is called a disease complex, and it requires a healthy American beech, which you guys have a lot of up in Western Pennsylvania. And it also involves a beech scale, which is this really small white organism here. And this insect will stab into the tree and cause a damaged bark enough that fungus can get into those holes, and this neonectrius fungus can then start to proliferate and kill the tree. And you can even see this kind of depression here under the bark of the American beech. And eventually trees will look a lot like this. And it's because they have that initial little stab from an insect, and then the fungus can get in there and proliferate. And how we look at it down here in West Virginia, we map it out. And because the insect has to happen first, you can see this red area here shows where the insect is, but this is actually where bark beech disease is in the yellow, because both of the organisms have to happen in order for the disease to happen. So a management tactic is to kill the beech scale, so that way there's no chance for the fungus to actually have a way to get into the tree. White pine blister rust is something else you'll see in northern western Pennsylvania, and it is caused by a rust fungus called Cronardum ribicola, which kills 5-needle pines, and we have a couple of them on the East Coast, but the biggest host is our Pinus strobus, eastern white pine. And it's a highly specialized rust pathogen that has 2 hosts in rotation each year. And so for part of the year, it will cause damage on our white pines, but then it will also live in a different life stage over on ribes. And the common name for ribes are gooseberries or currants. So it's a small shrub in the understory, and the fungus lives on the underside of the leaves, but then it will get to its next life stage and infect our white pines and cause a lot of devastating damage, because it'll go after the trunk of the tree and cause these huge, massive galls of just fungal tissue that will kill branches, and it can entirely kill a canopy of a tree if it's on the main stem. And like I said, it causes minimal damage to our ribes plants, and sometimes we recommend removing those bushes from properties. But if you're up in the Allegheny and you have like hundreds of acres of this ribes, there's no way you're going to do it. So it requires a lot of diligence to manage and find this disease. There is a slight success story, because sometimes I get scared that people think I'm going to just talk about how our entire forest is dying. That's not the case all the time. But there is a disease called thousand cankers disease, which also involves an insect and a fungus that have to work together in order for disease to happen. And this worked to our advantage in this case. But there is a very small beetle. And when I mean small, I mean like smaller than the tip of a pencil point. It's a small beetle that burrows on the bark of branches, and on its body, it just happens to have a fungus. It doesn't do anything with the fungus. It doesn't care, and it probably doesn't know that the fungus exists, but it carries it through the forest, and it will inoculate trees and cause these little cankers or dead spots. And when you have a lot of these little beetles covered in fungus, they can create it that the entire branch of a black walnut is not able to photosynthesize anymore, and it dies. So again, it sounds scary, but wait, there's success. The beetle what was here in Pennsylvania, but then it died, and we think it's because of weather patterns. So there is biological constraints that can sometimes happen to these, and we can understand them and use them to our benefit in controlling a disease. So we still manage for this, like we go out and we survey 4,000 canker disease in years where it has weather that is prone to the beetle surviving. And this is a really extreme example of how insects and fungi can be so co-evolved together in the world of plant pathogens that the insect will actually have a specialized pouch in its body specifically for carrying fungus. So this is a newish disease that is not in Pennsylvania yet. It's also not in West Virginia, but it is in Kentucky and Tennessee. And the disease is caused by a fungal pathogen called Raffaella lauricola. So here's the fungal strands, and here it is growing on plates. Pretty indistinct white colonies. And here again is another super small little beetle called the red bay ambrosia beetle, Xyloborus glabratus. And as an example, this is actually a slice of their brain. So this is that little beetle's entire head that has been sliced. And this is where their mouth would be, where it has a letter F. And there's these purple spots. The purple spot close up here shows a mass of fungal spores that the beetle, imagine like a chipmunk, will put in its cheeks and carry it to the next tree. And this is because it absolutely needs the fungus to feed its babies. So this is a direct relationship of how a fungus gets to a new host. It's worked its way in coevolution with the beetle to take it to a new tree that's dying. And the reward for doing that is that it will feed the beetle's babies, because the beetle babies, which sounds funny, but the beetle babies cannot eat wood themselves. They need the fungus to break down the hard to access wood into simple sugars and carbohydrates that they can eat. So these larvae will crawl around in these tubes that their parents made and literally like puked up fungal spores for them to grow on. And then the babies will eat the fungus and survive. And they'll complete their whole life cycle in that tree as it's dying from the plant pathogen. And then when the tree is dead, the adult beetle will put it in its cheeks and it'll fly to a new tree. So it's really interesting, that puzzle I was discussing. You have to figure out the beetle's biology, the fungus's biology, the tree's biology, and what all happens when they're together in a certain ecosystem. It's fun, it can be scary, but it's a really interesting and crazy story every time. And so this is what that disease looks like if you are an avid hiker and you see sassafras, which is the main host of the laurel wilt fungus in our area. If you see sassafras trees with those cute little ghost leaves dying and their bark is streaking, you should tell Penn State Extension that you found this, and you can maybe save the other sassafras in your area, because professionals will come out and take care of the problem before it takes over. So that leads us to talk about what you can do about plant pathogens, because again, it sounds scary, but prevention is the best tactic in controlling them. And of course, the big poster for Don't Move Firewood, it transports tree-killing insects and diseases. So if you literally cut down a tree that has sassafras laurel wilt, and then you take it to Cook's Forest, you could potentially kill all of the sassafras in Cook's Forest. And this does matter. This has actually been traced back for several diseases and insects. In West Virginia, we did not have emerald ash borer when everybody else did. And then all of a sudden, it was in the center of our state. And it was tracked back to a guy who drove from Michigan, cut down dying dead ash trees in his yard, and drove down to West Virginia to go on vacation, and boom, all of our ash are dead. So that's it. We know who it was. He's not in jail. He's fine. But that main lesson of that is don't collect or transplant plant material. And this also goes for keeping your vehicles on paved and gravel surfaces to stay out of areas that are wet and muddy, because if we think about what fungi like, they like wet environments. And you might be driving on some dead or dying disease stuff, and if you travel with it, it could go other places. You can remove mud on your shoes and vehicles before traveling to a new area. If you really like hiking and you go on back roads, take your car to a car wash, put your shoes in the tub, spray them off. I do, because I go to these disease sites all the time, and that is enough to kill the fungi just using soap and water. You can clean your shoes, your bike, your vehicle before leaving the county, which is a really good parameter that land managers use to track these diseases, the county level. And of course, when we do find these things, we can do reactionary work. There's a lot of mechanical things we can do, or even using fungicides, but that doesn't seem to work in my personal opinion. Usually that's too late. So trimming off of infected material and burning it on the spot can actually save an entire tree and then save an entire forest. And of course, research is a really big component of this. Sometimes with my job, people will say, well, why isn't anyone doing anything? Why isn't Penn State or whoever, WVU, doing anything? And it's because they are doing something, but research takes a really long time. And sometimes the organism that we're studying needs to be incubated for enough time for it to do what we want it to do. So research is a very big, key, important component of taking care of these pathogens. And just to show you research that I personally did, but also that it's not all doom and gloom when I'm talking about plant pathogens, I'm going to share with you what I did for my PhD work, which was using a plant pathogen to control an invasive species. So this tree down here is Tree of Heaven, which is an exotic tree from China. And you can see it growing here on the side of a road. This is my family's farm. And it really likes these disturbed areas like this. And this is a map. All the green areas is where Tree of Heaven has been introduced and is well established in our country. And again, this is from China, but it was brought over to Pennsylvania and Philadelphia, and it's spread across the whole country, and was also introduced around San Francisco, and has come across the whole country. But in 2002, in Pennsylvania, in Tuscarora, there was a patch of Tree of Heaven that were found dead. And it's because of a fungal pathogen. So we did Koch's postulate, which is that thing of where we take a sample, take out pieces of the wood, try to extract the organism, and put it back into a healthy Tree of Heaven to see if it causes the same thing. And it does. And so this organism is called Verticillium non-alfalfae. It is a plant pathogenic fungi that lives in plants that it's killing, but it also is able to cling onto soil and exist in the soil for a couple of decades until another plant that it can eat appears. So it has that ability, like the armillaria, to create melanized hyphae that it can exist and just wait for a food source to show up again. And it's really interesting because this fungus, in thinking about using it as a biocontrol, so many people were rightfully so scared that we would introduce a fungus that would kill the maple trees or that would kill the walnut trees or something else. But with modern day science, we can confirm through genetics and other protein analysis things that a certain strain of a fungus can have selectivity for a host. And what a strain is an organism is the same species, but a different strain. So you and I are both Homo sapiens. We're both humans, but you and I might look very different, right? I have brown hair, you might have blonde hair, brown eyes, blue eyes, that kind of thing. But a better example would be you might be lactose intolerant and I'm not, because that's a function of what we can eat. So this fungus, Verticillium non-alfalfae, there is a distinct strain in Pennsylvania that was able to eat and kill Tree of Heaven. And there are other strains that are on tomato, other things randomly. And you can see in this experiment, I took that fungus. So when I took the fungus from tomato and put it on Tree of Heaven, it didn't kill it. But when I took the Tree of Heaven strain and put it on Tree of Heaven, done, annihilated. And we can look at their genetics and see that they're very different, and they'll actually group in these really different clusters. So it's really exciting to have data that can show that these are different, a lot like if you and I were able to do 23andMe or some kind of other genetic thing that show that we have blue eyes, or part of us is from Africa, part of us is from Asia. And there's another success story, so again, utilizing plant pathogenic fungi in honoring your state tree, the eastern hemlock. So eastern hemlock, the Latin name Tsuga canadensis, is a beautiful tree that is native and encompasses a lot of the places in Pennsylvania that have a lot of cool streams, beautiful large trees that can shade out a canopy. You can see how dark it is here, because they're usually, or they're supposed to be, so thick and full of needles that can really shade out a forest and create a very distinct ecosystem. If you've ever gone hiking at Cook's Forest, the flora and fauna in that area are so different than the flora and fauna that are, I don't know, somewhere else in Pennsylvania. So this is what an eastern hemlock is supposed to look like. But because an invasive insect was introduced in the 1950s, the hemlock woolly adelgid, Adelgis tsugae, it caused a lot of our hemlocks to lose their needles to a point where they're very thin. And this is the insect down here. It sucks out the sap from the hemlock tree and causes the needles to die prematurely. And this is a photo here from Tennessee that shows a lot of hemlock trees that have actually died because of this insect. And so, like I was saying, this is a visual representation of what a hemlock forest looks like, right? It's cartoony, I know, but I made it a long time ago, but I like it. So in a hemlock forest, there's usually water. That's just a habitat type thing, but there's usually trout in that water because it's cool and it's shaded. There's also things like our trillium and our spring ephemerals that can be in that kind of environment that are very specific to the area, and it brings in a lot of wildlife and even distinct species of birds and insects that only associate with hemlock. Well, when the hemlocks start to drop their needles, you can see that the canopy actually will, or the forest floor will light up to levels of 70%, meaning that those plants that are adapted to shade will die. The fish will die. The birds have nowhere to lay their eggs. And so it opens up the forest floor for other things. Of course, the deer stays, but it opens up the forest floor for invasive species to proliferate, like our hay-scented fern, our tree of heaven, and things like our burning bush or euonymus. So it can really, this insect has a high level to like drastically impact the forest just by changing the ecosystem, again, like that American chestnut story. And what researchers or collaborators are doing now, like state management agencies, they're using chemicals to try to stop this problem. But that, of course, has a lot of problems in its own way. We know chemicals can be really harmful to animals and humans and salamanders because it's a chemical, and you need certain parameters to use it. There's also biocontrol methods of using other insects to eat that hemlock woolly adelgid, but they have their own limitations. So we were wondering, can we use plant pathogenic fungi to expedite management processes instead of waiting for these harmful or long-term projects to be utilized? And it's a really interesting concept where fungi actually have more than one job. They can be opportunistic pathogens, or they can be endophytes, things that are just living in plant tissue that are kind of maybe helping the plant to survive by trading sugars or some kind of like mycorrhizal situation. But usually they're just living as an endophyte or maybe something else in agreement. But if a tree gets stressed, there might be something that can act as a plant pathogen. And sometimes these plant pathogens will lay low, or they'll be only living in like the dead few needles of a tree. They're not going to wipe it out. Some diseases, just like us, you can have an underlying disease that doesn't kill you, but it is always omnipresent. So these are two examples of common plant pathogens all around you, like literally outside. They're somewhere out there. So this is a really distinct white colony with black spots. It's called Pestalozziopsis microspora, and it is a fungal saprophyte, but it can also be an opportunistic pathogen. And then there's another one that's beautiful and has these orange acerbuli that are goopy and full of spores, and it's Calototricum fiorinae, which is a fruit and harvest pathogen. Remember I said the thing growing on your bananas? That was a Calototricum. So it can act as a fungal endophyte. And that's what it was doing in this hemlock needle that I put on a Petri plate with agar. But there's been research that shows that these two organisms, given the right situation, can also eat insects. So they were found in the environment eating scales that were on hemlock needles and pine needles. And this Calototricum here has been found to eat insects when they are in the right environment. And so we took a bunch of fungal cultures that grew out of hemlock needles and put them on hemlock woolly adelgid and saw that some of them actually were able to eat hemlock woolly adelgid. And this took 2 years to do in many different states with a lot of different permissions, and it's still being pursued today in research endeavors, and it could potentially be something that's used against hemlock woolly adelgid in the future. So in conclusion, the field of plant pathology can be super depressing, but it can also have a lot of avenues that you can follow as a scientist. It is a dynamic and ever-evolving field of study that greatly impacts our lives every day, which might have brought your grandparents over here to America, which might have made your other family members not survive a winter in Appalachia because the chestnut trees were dead. And it also has the potential to make the world better, right? We can use it to our advantage to control invasive species or to maybe promote growth of something we want from a plant. That does happen too. And it's always super fun and always interesting, and there's always something new. And if you want to learn more about plant pathology in Pennsylvania, you want to report a disease or something, you can contact the Penn State Extension. It is their job to educate you. They also have a disease clinic where you can send samples, anything from the strawberries in your garden to a tree branch that's dead in your yard. And they will identify it for you and contact someone who can help you take care of it. The Pennsylvania Department of Agriculture, they have a whole page dedicated to different diseases in your state where you can read up on different pamphlets and PDFs all about the different diseases that you might encounter. And then also APHIS and the National Plant Board, if you really want to get super nerdy and read some scientific papers or something, they are key in controlling plant pathogens in America. And with that, I do want to, I guess, mention my Instagram. I usually don't, but I guess that's why I'm here. But if you want to find out more about fungi or just things that are in your forest around you, because I don't live that far from y'all, you can follow me on Instagram at K-Dubs the Hiking Scientist, where I post the fun things I find during field work and usually do a fun little write-up about them. So if you have any questions, I am going to stop sharing my screen and I will, I guess Stephen can probably unmute you all. Thank you for paying attention. I appreciate it.
Thank you so much, Kristen.
Thank you.
That was awesome.
So
yeah, thank you. I do have a question.
Okay, I'm ready.
Okay, I've got white pines, and the trunks look like somebody has spilled white paint down them. What's that? Do you know?
That is sap. Then that's probably from a bird or a beetle. So there's sapsucker birds that will actually go around and make little holes, and the tree they are drinking the sap, but the tree will continuously drop sap when they're not there. So you'll have these big streaky marks. And if your tree is sick, beetles will be attracted to it and drill into the tree, which also causes sap to come out.
How can I tell if it's a problem or?
If there are a lot of very small holes, like holes the size of your pen tip, those are beetles, and that's a problem. Okay. If they are little tiny boxes that are about the size of a dime in lines, then it's a sapsucker bird that is just trying to get the sap.
Okay. Thank you.
Yeah, and we have a couple questions in the chat that I was going to get to, and then I'll let everyone unmute and ask away. There was one really good question. You were talking about the fungus on Tree of Heaven. And there's a question here. that's basically asking, like, when you introduce a bioweapon, they put in quotes, how are you sure it won't kill desirable plants?
So that has been a big part of my career, and it's hard to say how long I've been involved with this because it makes me sound very old, but that fungus was found in 2002, and very rapidly it received a lot of money for funding because tree of heaven is such an invasive species and it's so hard to control that it got funded for a lot of research. And there were a lot of people working on it and doing all sorts of research questions, and one of them in the beginning was a host study of native trees that were in association with tree of heaven in the forest to see if it would jump. And then it was expanded on many different times to include things like the ornamental cherry or the Kuza dogwood that's growing in your yard. So it was very intensively studied by many different individuals, including myself, that did a lot of tests, both physically in greenhouses, and then also we did a lot of genetic work with the ability now to actually sequence the genome. We were able to find the exact thing that made it able to kill tree of heaven and not kill anything else.
Very cool. All right. So yeah, now I think everyone should be able to unmute, and if there's any other questions, I'm looking through the chat here. Yeah, K-Dub's the hiking scientist. I think that was put in the chat. Awesome.
Yeah, thanks for the great presentation. And I can say that the woolly agrids are here in Allegheny County.
Yes, they are everywhere in Pennsylvania.
Yeah, so I saw them about 3 years ago the first time, and I did a treatment on my hemlock trees, and it went away, and then they're back again this year. So you have to keep treating.
The treatments last about 5 years. They say 3 years, but not everybody's made out of money, and that chemical will last for 5 years. So that's what we do. I mean, just because also we are 30 people who manage an entire state. Pennsylvania has a little bit more people because you have more money and bigger area and more sensitive areas with the hemlocks. But usually we're on like a 5-year rotation for putting out that chemical because of limitations of being a human and money. So funny that we make these constraints on our life with money. We made all those numbers up.
And the other thing, I found a rust that infects Trillium. It was originally reported down in Westmoreland County, Pennsylvania. And I'm just looking at the paper. I think the paper was 2018. So I found it up in Allegheny National Forest, and it's on one of the other plants that they reported in the paper. So it's kind of interesting. I don't know if it was always there or if it's moving north. But I guess that's what we're going to start to see is things move north as things warm up a little.
Correct. On both accounts. So likely, I mean, you have a large member number here, but even within that, a lot of you probably never even heard of rusts. So just think about the 20 people who know what a rust is, and also the 20 of them who are crawling around on all fours in the forest to look at rusts, because the rust was underneath the Trillium leaves, right? Usually on the May-apples, they're underneath, the Jack in the Pulpit, they're underneath. So it takes a special kind of person to find something. And then we are also very lucky now to have things like iNaturalist, which you can instantly find out the range of something and if it's rare or not rare. And a perfect example of that is there is an extremely common beetle throughout the entire East Coast. And literally on a summer day, you can go out, put your hand up in the air, and you can like 50% chance you're going to have one in your hand. They're very small, but it has 7 observations on iNaturalist. But it should be in every single county on the East Coast. So we're just not there yet, and people just also don't know what they're supposed to be looking for.
I know there's on the beetles, there's a DNA barcoding project starting up from Cold Spring Harbor.
Yes, yeah. Beetles are the easiest thing to do DNA sequencing on because we've understood those primers for a little bit longer.
No.
Nice. There's a couple other questions in the chat here.
Please read them to me. I'm not good at reading.
This one says, why is the tree of heaven fungus not being more widely used to get rid of tree of heaven? Yeah, when you were talking about that, I had never heard of it before. And we have loads of tree of heaven here in Pittsburgh. Is that a newer management technique? Is it not available commercially yet?
So the earlier question asked the research question of how do we make sure? Well, that's 20 years. So a lot of these things take 20 years for us to make sure we're not going to kill the whales. And it's just what's been happening. And this year, and with me, it's so interesting because I've been involved with it for almost the whole time, like three quarters of the time that it's been around. And it just this year got funding from the Forest Service to be studied on how to be commercialized. So when I, hold on one second, stay right there. I'm gonna block it for a minute. Okay, I'm still here. I just had to stand up and get something. Okay, so the way that we grow Verticillium non-alfalfae is on these plates. This is it. And I'm gonna put this in your state next month. So it's coming, but I have to make this. So it's really, it seems easy, but it's really hard, and it takes a skilled person to be able to isolate it and to keep it in a lab and to grow it out. And so someone who works, and I'm not being insulting to anyone who works for a state agency, someone who works for a state agency either doesn't have the facilities, they don't have the skill set, or they don't have the time to take care of this. And so, because if you make this into what we call an inoculum, which is literally just the spores that we're putting into the trees in a liquid form with peptone, if you don't refrigerate it and you don't use it in 3 days, it's worthless. And it'll take probably this 2 more weeks to grow in order to be the right amount for it to be utilized. So it's hard for people to use, and it needs to be studied, which I did for part of my PhD work, for ways that land managers like PAGCNR can like drive in a truck and throw it out the window, or dump it out of a plane without having to have a lab. So there currently is money that was dedicated, like $300,000, to someone in Florida who has a biocontrol company to make a formulated way for this to be distributed. So likely within 3 years, people like PAGCNR and maybe even arborist companies could buy it and then put it in the forest.
Hey, we got a lot of cultivators here in this group.
Yeah. Well, there's also the legality of it still, because even with all that research, it's like people's names are on the paper that like, we're not liable, but like it can be traced back to why it's here and what it's doing. And it was also naturally found, because people started looking for it then, it was also naturally found in Ohio and Virginia, but has not been found in Maryland, New York, or West Virginia yet. So there's rules about what is considered to be native. And because West Virginia doesn't seem like it, but West Virginia is different than Pennsylvania. So on like a genetic level of the forest, we don't want to accidentally kill something. So there's a lot of research that goes on, and this stuff is actually being included in a research project with the USDA in Eastern Pennsylvania. And I have put the fungus everywhere I could already. So if you ever see dead tree of heaven, it's likely I was there.
I'm so happy you addressed the issue on making sure you're parking in the right spots and your footwear is clean.
Yes.
I remember writing a post a year ago about that, about how important it is to make sure before you go, especially within different watersheds, even different parks. Mm-hmm. Not only can you transport different seeds that are bad, but yes, fungus and algae and different things like that can be easily transported
mile a minute.
That way.
Yeah.
Yeah.
Yeah, mile a minute is terrible. Yeah, and Stiltgrass is terrible in Pennsylvania.
Yep.
And it can work. It's really simple. I throw my shoes. They're out in the yard drying right now. They were in the tub. But oh gosh, where was I going to go with that? Yeah, the spread of, I remember. So that site actually in La Crosse, Wisconsin. So with the American chestnut, it actually didn't have any chestnut blight at all. And then researchers went there to see what was going on, and someone had it on their equipment that they didn't clean, and then it was introduced. And so they were like, okay, now everything's changed. Now this is where we're going to put the virus, because now it's like a new study site. So even scientists do it. And I get it. People are really busy. They think it's not necessary. You're trying to meet deadlines and you make mistakes.
I'd feel terrible if I knew I was the person that transported like beach blight to a new location.
Can you imagine if I did something? It'd be terrible.
Yeah. Well, do you ever go? I mean, you were talking about iNaturalist. Do you ever go on there to find new, like, oh, hey, this was not recorded here. Oh crap, this person.
Yeah. So iNaturalist is in an agreement with the federal government where they have there's a group called EdMaps, which you guys can access for pictures. You can look at the maps. It's all open, everything like that. So any invasive species is logged in EdMaps, and it's taken from iNaturalist. So the observation is then accessible to people like me who work for the government, who can like actually control these things. And the thing about EdMaps is it uploads the things from iNaturalist. You can also directly upload to it, but professionals and experts that study bugs will confirm the identity. So it's not just like a random thing or someone taking a picture of like a picture and putting it on there, which does happen a lot, which is strange. I think people are trying to get like clout or something on iNaturalist, like because you get awards if you get most species or something. So it'll actually have people who go and verify its identity and stuff like that. So EdMaps is utilized by land managers and the federal government and state governments to find new insects. And like people even get alerts. So if something shows up, it's a lot like a Google alert. They'll be like, this is a new occurrence of stiltgrass in Allegheny County, and you'll get an email.
Interesting. Very cool.
So going back to cleaning shoes, in Australia 15, 20 years ago, they had sort of dips at the entry to each park to basically stop fungal or other things being traipsed around. And so the first thing you had to do is you got out of the car, and then you had to put your feet in these little bowls, which were refreshed with this, some sterilizing solution. So it's interesting, but that's the way they had to do it.
Yeah, they did that for white nose syndrome for the bats, because that was a fungal infection. And they're also doing that in Hawaii for the Rapidohea death, which is caused by a fungal pathogen. And it's contained on certain islands right now, but if you go to different islands, you have to like wash your shoes and all sorts of stuff because it's killing their native trees.
Fascinating stuff. There's another question in the chat here. Someone was wondering if you wanted to talk about spotted lanternfly at all, particularly because we were talking about Tree of Heaven as its host. And I do know there were a couple findings in Pennsylvania of some different fungal pathogens of spotted lanternfly.
Yes. I have to turn off my camera again. I'll be right back. It's right here. Don't worry. I'm self-conscious about everything in my life. Okay. So, so here actually is spotted lanternfly with the fungus on it. And so that is Beauveria bassiana. Oh, I'll put that in the chat that you're saying. She's a very nice girl. I'll figure it out. But this is a fungus that is actually used in commercial settings for greenhouse control of whiteflies and aphids. And there's a researcher at Penn State, Dr. Nina, I think her last name is Hajek, and there's another woman at Cornell, and they're both studying the fungus that was found on spotted lanternfly. So you can see the fungus pouring out of all the little like crevices in the spotted lanternfly's body. And They tried to do an aerial application with a plane last year, close to Reading, Pennsylvania. So that's East Coast. And it did work, but this again is something that research is probably going to take more time. And we also don't understand like how long the spores last in the environment. We don't know how much we have to put in the environment for it to be effective against the masses of spotted lanternfly. And there is another fungus called the Batcoa. I've never heard of that genus before this, but it is also a native entomopathogen, and they are generalists. So that means they're not specialists like the tree of heaven biocontrol fungus. They could potentially hurt our other native plant hoppers and tree hoppers and something else. Like I said, Beauveria bassiana is used in greenhouses against whiteflies. So it even goes on to other orders of insects. But does anyone remember the gypsy moth?
Yes.
Yeah, so the gypsy moth, remember it was so bad you could like hear the poop falling? I remember that. And there were no leaves in the canopy. Well, do you have a lot of gypsy moth now?
I don't think so.
No, you don't. Successful government treatments.
But it did harm some of the native moth species, those treatments.
It did. But there's multiple things that were used for gypsy moth, and they all still exist, and they're all still working on it to control it. So there was a virus, there was a bacteria, and there was a fungus. And there was also herbicide, and there's also pheromone stuff they do to the insect. But the fungus, Entomophaga maimaga, it was released ages ago, like decades ago. And then all of a sudden it had enough of a reserve in the environment that it was able to actually control gypsy moth. And there are, some casualties of our native population stuff, but it was almost like, well, do we lose our entire forest or do we lose these 3 species of cutworms? So there is, a trade-off. There's never like, oh, there's absolutely nothing that'll be killed, and that's something that they're considering with the spotted lanternfly entomopathogen. So there's a lot of research going on still with that, but it could be good, but it literally might take 20 to 40 years for it to work. I don't think so, but it could.
Fascinating.
Your tax dollars going to work.
Yay.
You may have touched on this before when I stepped away for a moment, but whenever you're traveling around, what's the best way to clean like your shoes and your clothing? And
I just use dish soap. I just put dish soap like this high in my bathtub, and then I put my boots in it, and so like the bottom is covered, that's all muddy, and then I rinse them off and I let them dry out in the sun. Another thing that fungi really like is moisture. So they're not in an environment on your shoes where they're growing, right? So they're like in this like in between stage of existing. And if you put them out in the sun, they will die also because you first use the soap and then they're getting blasted by UV.
I use a bleach spray and then I scrub them off and then I let it sit outside. So it's like a triple kill.
Yeah, nice.
I don't know if you go to visit some greenhouses, when you're going from house to house, they have these little pans that you step in, before you go into the next one.
That's exactly it. They're probably using something similar to a bleach. That's not just a soap.
Thank you.
Yes, thank you.
Kristen, thank you. That was a great presentation.
Thank you. I appreciate that. Thank you for coming.
Kristen, Stephen didn't ask my question. Where did you get the earrings?
I'm currently looking for her right now. It was a person who offered to make one off of one of my pictures, and then I ended up paying her because they were so good. But her name is Christina, and she's a very nice, normal person who just does art. Oh, I see. My internet's having a hard time on my phone because I'm on the internet on my computer. I'll find her and I will type her name in there. It might take me a long time to find her. I'm sorry.
Okay, take your time. It's okay.
I can email it.
Yeah, that's fine.
Thank you.
Yeah, if you email it to
Okay.
Julie and myself, we can get it to Cecily.
Okay, I will.
All right. Any other questions before we jump over to our mushroom ID session?
No, when we do that, if I can figure out how to show you my INET pictures, I can show Kristen the rhizomorphs I found today.
Oh.
Yeah, I want to see him.
Yeah, how do I do that? Just post that link. Let me try this. Let me try.
Yep.
Hold everything. Let me go back. Don't tell me.
All right. Well, if there are no more questions, then I will end this portion of our meeting. Thank you so much, Kristen, for being our speaker tonight. Really appreciate it. And yeah, definitely going to be on the lookout for more plant pathogens now.
Oh, you'll start seeing them.
Yep.
Yeah, thank you, everybody. I had a great time. I am going to stay to look at mushrooms with you.
Yeah, I just pasted my link, so maybe you can see it.
Oh, yeah, there it is. Thank you, Katie. That was nice of you. Oh, there they are. Perfect. Yes.
And there's a second photo if you click on the second one.
I'm inspecting.
And you